On August 10th, researchers at the Institute for Basic Science (IBS) announced findings from their work on Sindela, an experimental cancer treatment designed to target mutations present in cancer cells. The team found that the technology could kill cells from more than 10 types of cancer and slow tumor growth in mice implanted with cancer cells. Today, we examine the scientific principle behind the technology. How can only cancer cells be selectively killed? The reason it's normally hard to precisely identify cancer cells is that they closely resemble normal cells. Cancer cells arise when mutations accumulate in what were originally normal cells in our body. That's why attacking cancer cells can end up damaging normal cells too. In fact, some chemotherapy drugs affect normal cells, such as those that produce hair. So it's necessary to find a difference that appears only in cancer cells. Sindela works by comparing the DNA of normal cells and cancer cells, then using gene editing tools to attack mutations found only in the cancer cells. As cancer cells form and grow, letters that make up the DNA sometimes get newly inserted or deleted. These are called insertion-deletion mutations, indels. The research team selects the indels that are absent in normal cells but present only in cancer cells and targets those. Once the target sites are identified, the researchers use a guide RNA designed to locate that position together with Cas9, an enzyme that cuts DNA. If the guide RNA is the navigator, Cas9 is the scissors that cut at that spot. Together, these two make up the CRISPR-Cas9 gene editing tool. When the gene editing tool enters a cancer cell, it cuts out the mutated gene. In simple terms, it uses the genetic traces left by mutations as a signpost to identify and attack cancer cells. A different cancer for every patient, creating my own chemotherapy. The site Sindela needs to target vary between patients, because even people with the same type of cancer can have different mutations in different locations within their cancer cells. Currently, doctors test a patient's cancer genes and, if a particular abnormality is found, use a targeted treatment against it. But patients whose tumors do not carry that specific abnormality cannot benefit from this approach. Sindela creates a new guide RNA tailored to each patient's cancer cells. The gene editing tool itself stays the same, but where it cuts differs from person to person. Sindela's effectiveness and safety in humans, however, have not yet been established. Further testing will be needed before it can become a treatment, including studies to determine whether it works safely in people. Still, the technology points to a new possible route for treating cancer, and its progress through preclinical research and clinical trials will be worth following.